This is FX Medicine. I'm Andrew Whitfield Cook. We're at the 2019 UIC Medicinal Cannabis Symposium, Tweed Heads, and we're speaking with Dr. Scott Smid. Scott,
got it right.
I told you I would stumble over it.
No, you're good.
Scott's a pharmacologist working at the University of Adelaide, and you're particularly interested in the possibility of cannabis being used for various conditions affecting the gut.
Yeah.
Give us a brief synopsis of your talk.
Well, this morning I gave a presentation on some work that we've done over the last few years looking at how cannabinoids can influence gut inflammation. And so what I was able to do was develop a gut explant, so a tissue culture model, looking at how pro-inflammatory molecules can really damage the gut. And I'm sort of able to use that model to really then help understand how cannabinoids can modulate that inflammation. So I use pro-inflammatory molecules to generate tissue damage, and then I'm able to test a range of different cannabinoids as well as plant extract to see how they influence the progression of that tissue damage. So I'm using it as a model to better understand how cannabinoids might provide an immunomodulatory role to protect the gut from a whole range of different conditions associated with inflammation.
And I'd imagine, obviously, these are accepted pro-inflammatory cytokines, TNF-alpha, interleukins, that are seen in various disease states?
Yeah, no, they're very common and extremely pro-inflammatory in IBD, so TNF-alpha, interleukin-1 beta, IL-17A, so they all produced a consistent tissue damage in the human colitis tissue, and those were abrogated or mitigated through the endogenous cannabinoid, anandamide, and also by cannabidiol. And we did other research in using a range of different pharmacological compounds that block the CB1 or activate just the CB1 or CB2 receptors to help work out the particular CB receptors that were providing that benefit against the pro-inflammatory cytokines evoking damage.
Okay, so we've spoken previously to Morgan, who's a patient who has Crohn's and has had a horrible journey, particularly with medicines. They just haven't worked for her. And in her case, it was cannabis juice that was really working well for her and indeed gave her a new lease on life.
Yeah.
When you're talking about at the tissue level, I get the issue of control. We need to tease out sort of what molecules are active and which molecules maybe are useless. But then you've got the real world application, and in this instance it was the juice, the fresh juice. So how long does that trial and tribulation of what works, what doesn't, when you've got a plethora of molecules to deal with in the cannabis plant?
Yeah, well, it's very difficult. But the thing that was really interesting about the Taylor's case about Morgan was that the juicing retains some of the original progenitor acids and the THCA and CBDA.
Yeah.
And so, and Ian talked about this as well from the Lambert about the potential that the THCA or the CBDA might be providing the anti-inflammatory benefits. And so that's a big deal for someone like Morgan, because if someone with gut issues is taking tinctures and oils, that can exacerbate the condition because the gut can't really process those oils very well. So the ability for her to juice that extract was also quite important, not only in retaining some of these molecules that are lost when you have a conventional preparation where you're heating the extract to produce the neutral phytocannabinoids, but also the fact that she could tolerate the juicing much better than she might be able to do with a tincture or oil. So it had a double benefit. I was really surprised when I talked to them, as I had met them for the first time at this meeting, just the fact that she'd been able to go off some of those medications that just weren't working for her and that were having more side effects than benefits. So for her to just be on that cannabis plant juicing, she does need to juice a lot though, so she does need to take a fair bit to get control of her condition, but she was really at the severe end of the spectrum for inflammatory bowel disease.
So what do you think the options will be in the future? I know this is crystal ball gazing, but when you see something like that using a fresh plant, using the juice, which is obviously nutritive as well as having the chemical active components, the anti-inflammatory components, where do you think we're going to head? Do you think we're going to have, this drug for Alzheimer's, that drug for chronic pain, this one for glioblastoma? Do you think we're going to have a whole sector of various drugs that are high in this, low in that, moderate in that? Or do you think that we need to just use the plant and use the ancestral wisdom, if you like?
Look, it's a difficult question, and I think they can probably coexist to some extent, because I think you talk about the entourage effects and all of these chemicals that have anti-inflammatory actions, the terpenoids, the cannabis flavonoids, and they're all in small amounts. But if we talk about this entourage or synergistic effect, where 1 + 1 = 6, 7 or 8 rather than 2, there will certainly, I think, be a role for some of these standardised plant extracts that we can tailor towards particular conditions for gut health, especially the anti-inflammatory conditions. But there are quite a few different cannabis phytochemicals that have anti-inflammatory actions. So it might be the sum of all those parts, and that might be difficult to replicate within formulations that just contain maybe a small number of particular bioactives. But I think there might be a role for some of those smaller handfuls of bioactives together in pharmaceutical formulations for those that just may not be comfortable with the whole plant extract for perhaps a variety of reasons.
And what about with garnering funding for your research, getting approval? I would imagine at this stage it's cell work, so it's not subject to ethics issues. But with regards to just slogging away at research, yet having this spectre of its research on an illicit drug, how does that work? How do you...
Oh, look, it's prior to the last couple of years with just the advocacy and the opening up of access, it's been very difficult, and that's the same not just for Australian researchers but for researchers in Europe and the US as a Schedule 1 in the US. So very hard to study the plant extract up until just recently. So I can obtain plant material through distributors and through, say for example, with a Bedrocan product, so I can obtain that, extract it and use it for plant extracts right now with the necessary permits, which we have in our department as a pharmacology department. So we can extract that plant material and characterise its bioactivity, but not everyone has that capacity to do it. For the human research studies, even though I'm using biopsy tissue, always need to obtain Human Research Ethics Committee approval for use of that human tissue. So we always have the ethical constraints or the limitations to overcome. Sometimes that can be kind of onerous, especially when you're just working with human tissue. I'll go up to the patients that are going to surgery and I'll ask them, I need your permission or informed consent to provide this tissue, and they'll go, well, you're welcome to have it. There's not much I can do with it now. It's when it's coming out of me. So there are some practical logistic difficulties in doing these types of studies, but I think the benefit it provides for me is well worth the motivation. Funding cycles are no different for many other Australian researchers. I'd love
to slog.
I say to some of my people in the talks, when I'm giving talks in specialist groups in Adelaide, I say, if you know of any wealthy pastoralists or mining magnates in South Australia that want to give me $30 million to set up an institute, I'm more than happy to. But that's kind of, the exception rather than the rule. And mostly it's applying through government funding circles, but also industry links. And that's where coming to the UIC Symposium can really help provide some of those links as well. So that's been quite beneficial.
And talking about industry links, we've spoken about this ad nauseum, about cost and patent and ownership of a plant that's been used historically for eons. Where do you think this is going to end up? Where's your crystal ball leading you?
Oh, look, I couldn't tell you, to be honest. It's very difficult to get a feel or to prognosticate on what the end point is going to be in regards to IP. A lot of the companies often look at patentable methods for application or delivery rather than plant products. So I think that's going to be quite a challenging environment. So it'll be interesting to see what happens down the track in industry as a whole, not just with IP, but obviously the business model, the gatekeepers for provision will be clinicians. It's sometimes a bit of a battle to see how the clinical trials will go, what evidence you'll be able to provide to give them the confidence about prescribing for their patients. If you can get that, then I think the supply bottleneck, the access to patients, will be a major hurdle that can be overcome. And the research all helps contribute to that, whether it's preclinical or clinical research.
Last crystal ball question is, we've spoken about access and your preclinicals. What do you think, with your expertise talking to other researchers around the world, not just Australia, What do you think the timeline is going to be from preclinical to finished product to using it on the market for the benefit of the patient?
Well, I think it's a lot less than conventional small molecule drug discovery and development, which will give you a standard timeline of
The molecule's been discovered.
10 years, yeah. The molecules have been discovered. There's a lot of them, and we're still trying to work out all of the combinations, but I think it'll be a lot shorter. I think that the time frame for these types of studies will be in the order of 4 or 5 years rather than decades. So I can see a short-term horizon in terms of significant breakthroughs in that area.
Shorter rather than longer, please. That'd be lovely.
Yeah, absolutely.
Dr. Scott Smid, thanks so much for joining us on FX Medicine.
No, thank you. You're welcome.
This is FX Medicine. I'm Andrew Whitfield Cook.